Hundreds of ejaculates of different concentrations are used to perform the calibration. Unlike most photometers, the spermacue directly provides an output reading in sperm cells per milliliter. First, turn the unit on and pull the black slide out of the lower right-hand corner until it clicks, with the chamber for the cuvette fully exposed. The spermacue will reset itself to 0 each time the slide is pulled out to this position, and then in a few seconds the display will read "Ready." The raw ejaculate should be mixed gently but thoroughly to ensure that a representative sample is obtained. With a pipette, a sample of semen is removed from the ejaculate and the cuvette loaded by placing a drop in the open end. This requires less than 1/2 milliliter of semen. Then the cuvette is wiped on both sides with a lint-free tissue, taking special care not to touch the open end. The cuvette should be checked to ensure proper filling without any air bubbles. The cuvette is then placed carefully into the holder and the slide pushed into its closed position. The unit will display "Measuring" in the LED display. The unit takes several readings, and after approximately 15 seconds, the result, measured in millions of sperm cells per milliliter, will be displayed. All photometers have a range in which they are most accurate. Therefore, if the resulting spermacue reading is greater than 450, an underestimation of the actual number of sperm per milliliter will be obtained. In this case, the ejaculate should be re-evaluated after first making an accurate dilution. The dilution is prepared by pipetting 1 milliliter of 2.9% sodium citrate solution in a new test tube and adding 1 milliliter of raw semen into the same tube. Gently mix and load a cuvette with this solution. The resulting reading from the spermacue must be adjusted to account for the dilution. The adjustment is made by multiplying the reading by the factor of 2, since the dilution represented 1 part of semen plus 1 part of sodium citrate, or a 1:2 dilution. The spermacue is most accurate in the range of 150 to 450 million sperm cells per milliliter. Under 150, error in the estimate will exist, but this error will be less than if high readings over 450 are obtained. Only a few insemination doses will be produced from ejaculates with very low sperm cell concentrations. Total sperm in the ejaculate is again calculated by multiplying ejaculate volume by concentration. The spermacue is supplied with a red control filter micro cuvette. Each machine has a unique control filter and corresponding filter reading by the spermacue. This filter should be used daily to check the operation and cleanliness of the optics of the spermacue. The calibration curve is permanently burned into the chip of the spermacue. The unit does not have a light bulb, but rather uses a light-emitting diode as the light source. When necessary, the user can easily clean and adjust the spermacue. Semen evaluation. Microscopic evaluation of sperm cells for motility and normal structure, morphology, is a critically important step of semen processing. Microscopic motility and morphology estimates of semen are the most commonly used quality control procedures. However, it is important to remember that the correlation between these estimates and fertility is relatively low. This means that even with high sperm motility and a low abnormal morphology, fertility may be impaired. The initial semen evaluation should be done at either 100 times or 200 times total microscopic magnification, where a fairly large field of sperm cells can be seen, and then a second evaluation should be done at 400 times magnification to determine morphological quality. A third evaluation at 1,000 times magnification may also be done to determine the acrosome integrity of individual sperm cells. A heating system for the microscope stage should be considered a requirement for use during motility evaluations. All laboratories processing semen should have a slide warmer to pre-warm the microscope slides and cover slips and to keep them warm while preparing semen samples for evaluation. Raw, undiluted semen should not be evaluated, as it is virtually impossible to make an accurate assessment of semen quality due to the high concentration. Likewise, the swirling motion observed in raw semen samples does not indicate motility of individual sperm cells and should not be used as an indicator of quality. Virtually all boar semen samples must be diluted with an extender prior to evaluation of motility. Place a drop of extended semen on a warm slide and place a cover slip gently on top of the drop. Immediately place the slide under the microscope and focus on the sperm cells under either 100 times or 200 times magnification. Observe the movement of individual sperm cells and make an overall evaluation of the ejaculate, giving it a quality score indicating the percent of motile sperm cells. Be realistic about your evaluations and the motility scores you give the samples. Now look for abnormalities of individual sperm cells, such as proximal and distal cytoplasmic droplets, deformed heads, and bent or kinked tails. Sperm cells with these abnormalities are generally considered to be incapable of fertilization, as are non-motile cells. Again, give the sample a score. Next, a composite score should be given, which takes both motility and morphology into account. Sperm cells which are considered non-motile and have a morphological abnormality should not be considered twice, as your goal is to give the sample a score which indicates the percent of normal motile sperm cells. Because of the relative low correlation between motility, morphology, and fertility, and the inability of the human eye to accurately assess motility, the industry has generally accepted that a minimum motility cutoff should be approximately 60%. Ejaculates with motility less than 60% should not be used. The number of normal motile sperm in the ejaculate is determined by multiplying the percent of normal motile sperm cells and the total number of sperm in the ejaculate. For example, if the motility score is 95% and the morphology score is 90% normal, the composite score is 0.95 times 0.9 or 85% normal motile sperm. Then, the number of normal motile sperm in the ejaculate is the total sperm in ejaculate times the composite score. For example, 96.7 times 10 to the ninth total sperm times 0.85 equals 82.2 times 10 to the ninth total normal motile sperm in the ejaculate. To determine the number of doses of semen that can be made, the number of normal motile sperm is divided by the number of sperm desired per dose. If, for example, 3.5 billion normal motile sperm will be in each AI dose, then 82.2 times 10 to the ninth divided by 3.5 times 10 to the ninth equals 23 doses, which can be made from this ejaculate. Once the number of doses of semen to be made has been determined, the next step is to add the correct quantity of semen extender to the ejaculate. This can easily be done by measuring the volume by weight as the extender is added to the ejaculate, or with the assistance of a peristaltic pump from a large extender stock in a water bath or from a large vat of extender. Adding the extender to the ejaculate can be done in one step. You should always add extender to the semen to prevent dilution shock, and do so slowly by letting it run gently down the side of the container into the semen. Once the ejaculate has been fully extended, it should be gently but thoroughly mixed. A sample